And then I would also think about a way to make this very close, because I think one goal of the paper is to make this closer to what is known about the circuits. Because in human literature, many people operate at the brain-wide level, and they cannot touch too much the local and neuronal level, whereas in the mouse literature, this is much more direct. And so I feel like uh, one thing we could do is show real neurons on the right, and then in the middle, uh, show exemplar real neurons, as given by the EM microscopy, or things like that. Just make it really anchor into the data. That's just one comment I might have. I'm curious what you think.
Yeah, that's a good idea. Wonderful. I'll maybe try and
So
come up with a couple drafts.
I have similar impressions. So for example, for the real neuron, the diagram we already have in the original manuscript, figure 6, for example, and some spikes with that. And for the local circuit, also quite available in figures 4 and 5 in the original manuscript. So I think it's going to be quite, if you can align both the figure, the arts here, and also the color code, it's going to be quite cool.
Makes sense.
All right.
Any other comments for Chris? Or?
Jerome, I think we already have these on, we have this over like a Google Doc graph, right? Can we just share the link with him, and so it's easier for him to access them? These all the like earlier graphs we made.
Oh, okay.
Yeah, I think it's gonna be a lot easier than just copy pasting from the PDF. We can just have the original figures for him.
Oh, yeah. The figures of the manuscript. So we have a Dropbox with Adobe Illustrator files now, so we can share it. Yeah.
And another comment, I would keep the human brain. I'm not sure why we should change it to mouse brain. I think it's more impactful this way. And anyway, we think these computations are relevant to human brain.
Okay. Your opinion on that? Sorry, I just need to see everyone's. Oh, Alex, maybe said maybe a column for the middle.
Like a cortical column.
Oh, I see.
Yeah, because it's circuit. So somebody said if we show the neurons, then we kind of have to worry whether it's the predictions are on the top or the bottom, because is it the axon, or is it, maybe we can just get around it and show a microcircuit.
Yeah. I mean, we could overlay E and I cells with a representation of the layers, maybe.
Yeah, just some kind of network. I think people, Sarah made a good point that by showing the neuron icon again, people might take it too seriously in terms of where do the predictions go and where do the errors go. And in a way, we're abstracting away from that, right? So we're saying, well, it's happening in the circuit. So we just have to be careful with our graphical language, or we don't get too confusing or too specific.
I see.
Yeah, I mean, so one of the reasons why it's set up the way it is that it if you have the same illustration for all three levels, that it's nicely conveying the hierarchy or multiple levels. I mean, but I guess that if we maybe figure out a nice graphical way to capture this idea without cloning the neurons in the way it is right now, then we might be good with that. And then when it comes to public domain, these are just like placeholder images for now. I guess I took them from like an NIH repository, so it's perfectly fine in terms of copyright issues. But then I guess that these will also be polished down the road, right? Jerome?
So, so our plan is so Benedict Rossi has joined us today. She is a professional scientific illustrator, and our plan is to take this figure and make a polished versions with Chris and everyone, so that it, yeah, because this figure is really important. We want it to look nice and clear.
Yeah, just a quick, it's a good point about making it visually clear that there's a hierarchy or that it's iterative. So I do like that. So that maybe there's another way to address Sarah's point.
Yeah. Okay. So should we, maybe we'll summarize the feedback, Chris, and then follow up on the thread, on the GitHub thread, so that and then we can make a second draft with Benedict's help and yours.
Yeah, wonderful.
Okay. Any other feedback for Chris before we move on? Okay. Okay, so now I'll hand it off to Colleen. Hey, Colleen, are you here?
Yes, sorry, I was looking for my mic button.
Do you want to share your screen or do you want me to share the screen?
I'll share the screen. Let's see. Okay, hopefully it's sharing now.
Yeah, we see the Google Slides view.
Great. So I just want to start right before jumping in, just to let everyone know sort of what's needed going forward. What are sort of the last steps before we can submit? I tried to do my best to just compile this information since, you know, with 50 authors, you can end up in a position where everyone thinks someone else is going to do it. So I tried to just make sure the information is all somewhere. So we're working on the final graphical abstract, finalizing the figures, after which we'll have to revise the captions to reflect the updated figures. Then there's just the preparation of the manuscript for a submission, which is not too demanding with physiological reviews. We have a bit of an oddity that our captions are in the figures because of Google Doc. We'll have to see if that's a problem or not, but I'll assume it's not a problem until they tell us it is. I'm also missing author information. We made a table because, you know, 50 authors. If anyone knows the people I haven't been able to get a hold of, can you just please ask them to click on the link? I've just got no information yet for Roberto De Filippo and Martin Vink, and then just 2 columns missing for Simone Russo and Eli Senish. So please, if you know them, just send them an email. And then I'll just mention that we're going to want to decide at one point on the license. We do want to pay for the paper to be open access, right? That only makes sense. And then we have 3 options for licenses. And so, yeah, there you guys have that information. And now we'll jump into the main event, which is the figures. So basically, our rationale for updating the figures was they were made by different people, pulling from different papers, and so the styles were not particularly uniform. We thought, let's do what we can to improve clarity by making the style a bit more consistent, aligning basic things like font and sizes, but also how we use shapes and symbols, like for inhibitory and excitatory connections, to try to have the language as consistent as possible without going overboard and redesigning everything from scratch. So we kind of tried to find a happy middle ground. You'll see that it's not a completely consistent style, but we tried to make sure that if we use a color scheme, it was somewhat recognizable across figures. And we did check copyright and licensing for all the figures that are being reused. We got permissions from the authors where we needed permissions based on the license. And we also did try to double check with any of the original creators of the figures, because of course it's your work that we've sort of jumped in and modified. And so especially if it's your figure, please let us know if you don't agree with something that was done, because of course you conceptualized that figure. All right, I think that's what I had to say. Oh, and thank you for the public domain mention, Alex. I did notice that, yeah, the figure that we have that has an image was a public domain image, and I know now that that was not an accident. So that's really good news, because it's really bad when you get to the publication step and you can't publish the figure. So thank you for that. I've also tried to add that to every caption where necessary. Hopefully that will be all they need. Okay, Now, a lot of these figures were modified by Bénédicte. I don't know if we want to both go over them, or I can just give a quick overview and then Bénédicte can jump in if we have discussions about the specifics.
Yeah, that works. I follow the instructions mostly, so.
Yeah, great. So I don't want to take too much of people's time. I'm not sure because we have a lot of figures. Jerome, do you have a thought for how we should approach this?
Yeah, or I would just introduce the rationale for the change and then ask for questions.
Okay. Figure 1, you can see we changed the shapes so that they were squares, which is what we used later in other figures. We tried to align the use of, every time we try to represent oddball on the paper, we tried to use a reddish color. And then any other edits that were made were matched to a later figure, I think figure 13 or something that has similar color scheme. So this was fairly minor changes to this figure. Do let me know if you have any questions or concerns. All right, I'll just thank the people who have written to the people I'm missing. Thank you so much. Okay. Also, if you have any thoughts later, of course, please just jot them down somewhere. The second figure had much more substantial edits. So here we did change the color scheme. We pulled a bit from Laureen's figure, this one. So we sort of pulled some of the color scheme, the inhibitory being in the bluish region, and then the positive and negative prediction error neurons having this gold and pink colors. That was pulled from there. And then you can see just minor stylistic changes. We changed the ends to be upside down triangles for excitatory and flat ends for inhibitory. That was the convention that was going to require the least changes across figures while keeping them consistent. Another thing to mention is,
I want to highlight, so the panel B is very important. It's taken from Georg Kahler's and MC Flogel paper. The modification from the review is that we added connection between brain areas. So in the review, they only have one area, but they mentioned that they are doing hierarchical processing schematic. And so here there is prediction coming down and error coming up. So this is not in their diagram, but I think it's implied. And so to make the figure a little clearer, we only are setting up the negative prediction error. The positive prediction error are local. But I think it's implied that both will go up, but it just makes the figure very crowded.
Yeah. So that's something that we'll make sure to write in the caption. If you guys think it should be indicated in some other way, I don't know, a partial outgoing that sort of fades out. I don't know if you guys have any ideas, but yeah, we want to avoid over cluttering the figure. Also, we really do welcome people double checking these circuits. They've now been through a few iterations, and of course we want to make sure that there are no mistakes. There had been a sort of oddity in this figure, which, sorry, I'm now seeing some things that look new. So anyways, it will be great to have people just take another look, make sure that the negative error is dendrite or neuron is doing what it's supposed to be doing, the positive one is doing what it's supposed to be doing, and everything is connecting as expected. Any comments?
Question. So am I seeing this right in panel B, that the negative error is receiving the prediction after a negative sign? So it subtracts the prediction from the bottom-up input, right?
Right, because it's doing positive and negative subtraction. So you
it's
have
sensory minus prediction. So the negative error is doing sensory minus prediction, and the positive error is doing
Oh, do we have the color swapped?
Sensory plus prediction. The sensory. Okay. Yeah, if you could say, so what is it that the negative error is doing? It's receiving the bottom up and it's
The
receiving the
negative
prediction.
should be, I think it's doing prediction minus sensory.
Could we zoom on panel B, please? On the right column.
Colleen, can you hear us?
Sorry, I was just answering something in the chat. Sorry, what did you say?
So if you could, it's easier if you could zoom so we make sure we're looking at this correctly. Yeah, so I'm focused on panel B exactly. And then the computation that the negative is doing and making sure that it's right. So we're showing that it receives the bottom-up input and it receives the top-down prediction through a negative sign, through that interneuron. So it's doing sensory minus prediction.
That's right.
So, and that's what
we'll have to check our convention. Yeah, it could be swapped. I don't recollect what was the, yeah. You remember, Pauline?
This input should be going here. This blue input should be going here, right? Both inputs are targeting the inhibitory neuron.
Oh, we could just swap the colors.
So the dark blue input is coming from brain area 2. And you're saying, so that's the prediction.
Yes, it would be getting prediction minus sensory. Inhibited, yeah, prediction minus sensory.
Which one is the sensory? Which one is the sensory here? That one?
This one,
right,
which is hitting the
so
inhibitory.
Prediction minus, okay, right. So then I guess you're right that the dark blue from brain area.
No, but that's this one, Colleen, is the prediction. The sensory is coming from downstream. So that's the challenge here, is that we are wiring between areas, and then the sensory
this is sensory.
What?
This one is sensory.
That's prediction. That's an internal model. It's going. You see, it's going down. It's
the local prediction from the same area.
Yeah, we might need a because the problem is we have errors that are calculated locally and propagated up, and so they become sensory for the next area.
Yes, sorry.
But maybe the color scheme is the problem, but or we need to add a text.
It would help. I guess I had this comment on the earlier version too. It would help to write PSE on. That would really make it easier to follow. And then another question for the positive error. So there we are showing that it's going, the prediction from brain area 2 as well as prediction from brain area 1 are going there. So sensory is going through negative. So it's P minus sensory. And that is correct, I think. So I think the positive is correct. Or should it be sensory minus? Now I'm confused which one is positive, which one is negative.
It's a matter of conventions. We should check our glossary.
Right.
Karim?
Yeah, so why there's asymmetry between the negative error projection in model B to the pyramidal of the higher layer versus the positive error neuron projection to the interneuron in the higher layer?
Asymmetry. Well, because yes, Colleen.
Sorry, this was to avoid clutter, right? We wanted to indicate it in the caption that both of them are sending projections.
Yeah, I understand, but the figure makes it clear that the negative errors project directly to the pyramidals, whereas the positive errors project to an interneuron intermediary.
Oh, sorry.
Yeah, this guy goes through the interneuron, and then the interneuron inhibits the pyramidal at the higher layer.
Yeah.
Where this is not the case for the negative error pyramidal.
That's right. So this is the circuit that Kählers and Flogo propose, where the top neurons is an internal representation neurons, that becomes but is also a prediction for the downstream area. And then and as we said, the error goes up. So it becomes the bottom up from the next area. And because you have positive and negative errors, they have opposite influence on the internal representation. And so one has to go through an inhibitory cell.
But not both?
No, because the negative error is the right sign to correct. It's like you have a prediction and the negative error, and so the negative error is pushing you in the right direction. Whereas if you have a flip error, you need to flip it.
A break?
But follow up on that. Why is it that the negative error, the projection from brain area 1 goes to negative error? Yeah, exactly, that one. If you follow that, that goes to here, to negative error from brain area 2.
Yes.
But then the positive one does not project across areas and only projects locally.
That's right. That's something that I tried to write to draw with Benedict, because the way, and this is not in Björk Keller's diagram, because he just drew one area, but wrote in the caption that this is assuming errors are going up and predictions are going down. So you would assume that both the positive and the negative will go up. But if you follow this logic, the positive error should go onto the positive error neurons of the higher order area and the opposite inhibitory neurons. And so that becomes, yeah, you could draw it this way, but that just if you think
too
that's
many
important,
lines. It was getting unreadable.
It becomes very clutter. And then also a lot of errors occurred during this. And so I don't know if it provided clarity. If you think it should be added, we can.
Can you say again, what is it that should be added? And we didn't add because of...
So you see the positive neurons, the positive error neurons, in principle should project up. It should follow the negative error, but it will not go in the same place because it's opposite sign. And so it will have to go on the corresponding inhibitory neurons. So for it will go, because it's a bottom up. And so it will go on the inhibitory neurons of the negative error neurons and on the pyramidal cells of the positive error neurons. So because they are flipped, it becomes the signal. That's right.
This very artistic rendering would be sort of what we're missing.
So in the positive error neuron, the error is computed and then it's sent to that dark blue internal model neuron up there to update it, right? So that error goes...
The positive is doing prediction minus sensory. You see?
So yeah, and it goes up there. So I'm talking about the arrow up there.
But yeah. But if your prediction, if you have a sensory that comes up in two bundles, one is opposite sign of the other, they should not have the same projection pattern.
I'm not trying to understand why the positive goes through inhibition to get to that dark blue internal model neuron.
Oh, well, because it's trying to correct the prediction. It's an error signal. And one is going to correct the prediction directly, and the other is going to project the prediction with an inhibitory neurons because it's swap. It's their invert.
Yeah, but that's one mechanism, one possible mechanism. It could be done in different ways. For example, if one imagines that the negative neuron, error neuron, is projecting to the same interneuron as the positive error neuron, and it's basically one is pulling that neuron down, the other one is pulling it up, and then the differential of these two signals is what inhibits the blue higher level neuron.
But they can't act opposite if they're both excitatory neurons, and so I think that's why we end up using the inhibitories, because we're saying that all the triangles are necessarily excitatory. So if they both projected to the inhibitory, they would just sum.
I'm not sure I followed you.
Oh, maybe I misunderstood what you were proposing. I thought you were suggesting that, for example, the positive and negative could both project to the inhibitory interneuron.
Yeah, so so basically if we have an unbalanced input, both are excitatory, right? But one excitatory neuron, one of them, the pyramidal, let's say the negative error pyramidal, is downregulated because of the interneuron at the lower level. And then the positive error neuron is upregulated, right? So that would create a difference at some type in the mean average of the interneuron projecting at the higher level area, right?
But it wouldn't differentiate between which was higher and which was lower.
It wouldn't. Yeah, but I'm saying it's just going to create a differential signal. So what you're saying is that by separating these two couplings, the dark blue neuron at the higher level is able to tell which error type it's receiving.
Yeah, I guess
right?
it's able to be pulled in the correct direction is the idea.
Yeah. There are many ways to slice this cake. I actually, this is a proposal that Keller and Flogel made. I don't think it's based on very much. I mean, it's based on some knowledge of the anatomy, but that's mostly based on mechanistic and proposal. But I think there are many, many ways to slice this cake.
Yeah, that was my point. Is it an established circuit diagram that people have verified with, in slides or whatever?
It's one of the proposed diagram that had lots of citations.
Okay.
One suggestion about the diagram. So if we are showing the inter-area connections for negative neurons, I think we should show it for positive neurons too. It's otherwise it's very confusing. I thought we are trying to make a point here that positive doesn't go, but negative goes. But if the plan is to not have a very cluttered diagram, how about we only show inter-area connections and not within area, meaning that we remove the connections from negative and positive neurons to that dark blue internal model neuron within the area, and we just make it about inter-area. So at least we're consistent. So I'm proposing two. One is that we just have a full diagram and we put the arrows for the positive neuron inter-areas back, and we sacrifice how decluttered the diagram is. The other is that if we're going for a decluttered diagram, then we just keep the inter-area connections of errors updating internal model, and we remove the within area.
I mean, because I think the intent of this panel is to highlight one proposal that came from a group. And
then I would put the inter-area for positive neurons. That's just very confusing to me that we don't have that.
Yeah. I think I would if it would be great if Arzane, if you read Keller's piece. I mean, I'm sure you did, but reread it, because I think you will see that what we're trying to fill here is, I think, places where the review was not very clear either, and so I think that's just it's just the state of the literature is unclear on these things, and so I would.
Yeah, no, I remember their paper very well. I don't think I can reread it. I don't think that will benefit much. So I'm saying how to make this
we
clear.
can have the second one.
Or we can have, for panel B, we can have two versions. One that focuses on within area connections and the other that focuses on inter area. I think we're trying to fit in too many lines and arrows in one figure.
That's right. We could try. We certainly could had also a paragraph discussing those, because I think I know, as we said, there's many ways to do B, and we should just highlight this is one way. Yeah, I think Sarah said that too. Yeah, because I think I don't think we have that language in the review too much.
make a note for the caption. And then, yeah, the other idea, right, Fazane, you were suggesting that we sort of duplicate one with within and one with without, or between.
The
Which
one that
actually,
focuses on between area updating, the other on within area updating of the internal model.
yes.
And then the
Because
between
Sarah
area
proposed that we
updating.
do positive versus negative. But I guess the between versus internal advantage is we only need to show one area for the within. And then we can bring both areas for the between. Yeah.
I think we are thinking in the right direction to simplify it instead of eliminating. We break it into two figures, whether we do it like what Sarah is saying, positive and negative separately, or like what I was saying with within and between. I guess we can see.
Yeah.
Yeah.
Okay. Maybe, Colleen, we should move on.
Yeah, and I do want to reassure everyone that this was probably one of the most complex ones. So it comes early, but it does make sense that we spent more time on it. It won't be like this for all the figures.
Comment here, so I look at the look at
Oh.
the legend to find the paper because I want to give it
Sorry.
a read. It's referring to Keller et al. 20.
Oh, sorry.
Oh, sorry, you cut off.
Wait, yeah, I thought I look at the caption, so there's no paper Keller et al. 2021 in the reference list, and I think it's gonna be the 2020 A or B paper actually we're referring to here.
It was 2018. Thank you. I noticed the same thing. I just fixed it.
Amazing.
Yeah,
Yeah,
thank
because I
you for
want to give it
pointing
a
that
read and I
out.
didn't find it. So yeah, thanks for acknowledging it.
Yes. Yes. So it's Keller 2018, and it should
Thank you,
be the
Jean.
second figure. Okay, I want to move on, but I want to make a note for what Sarah said about mentioning the caption.
Yep.
I'll show you guys the next one whilst I do that. So the next one was a figure that we got from Campagnola 2022. They were kind enough to give us the vector version. And so we made minor changes. We kept the weights of sort of the colors and the size of the arrows were determined from data. So we wanted to keep that exactly. And then we just changed the style and the acronyms of the neurons to better match what we have in the paper. And I'll take any comments while I quickly jot down what I wanted to jot down. Caption.
What are the dots here on the left panel?
Oh, yeah, these were differences. These were connections that were stronger in layer 2-3 versus layer 5. And when we were checking with the authors, they said, oh, if you want, remove the dots because it might not be relevant to your use. So we removed them. So yeah, comparison layer 2, 3, and 5. All right, I think maybe everyone's okay with this. Moving on to figure 4. This one is from Jordan, so Bastos et al. Jordan Ham's paper. Oops, forgot to write. No, okay. So we made minor changes to the style for consistency. So we changed the colors of the interneurons to match what we use elsewhere. And then just a few stylistic changes. Everything else is held pretty much constant. I'll zoom in. So hopefully that's okay with everyone. For the next one, we made almost no changes. This is Lorene's figure. First, we don't have it fully vectorized, so we did keep it a little bit. So for example, this section can't be changed. But since we decided to sort of use this color scheme going forward, so it's already fairly consistent with the others. It looks a little bit different. I could make some changes here, like writing in PYR, which I haven't done, if people think that that's a good idea for the prediction error neurons. I just realized that I left that as is. Do people have any opinions on that?
I think we should do the same, like on the figure 2. Yeah, write down sensory input or like area 1 input, area 1 prediction. You see, like as she did, just to clarify how an error is becoming an input. I think that would.
I
narrative.
see. So brain area 1.
Input, yeah.
Okay.
But not for this figure, for the other one.
Oh, wait, sorry, what?
For the figure 2.
Ah,
Yeah, like
so we should
here
write
it's-
prediction and sensory.
Yeah, I think the same way as she did here. Yeah.
Got it, yes.
That will clarify.
Okay, sorry. I
Don't worry.
Okay, the next one we have is Fabian's figure here. We really just changed the color scheme primarily so that it matched somewhat to the Lorraine's paper, with the predictions being green and then the sensory input being this orangey color. Now that's obviously something that we don't represent in figure 2. This is where we start to meet the limitations of using a color scheme when the figures show overlapping but different concepts. Please let me know if there's something that you think we should change here. We also did go in and change the ends of the inhibitory connections to be flat heads. I know that they aren't as visible. Do let me know if you think this just doesn't really work. All right, next, this was Jeff's figure. And what we did here was we just changed the color scheme, since we've sort of reserved red for the oddball. So flip the green and the red. We did check that they're mostly colorblind safe, the colors. Actually, they are colorblind safe. And then we also decided to change this from a set of colors to a gradient, just for a little bit of easier interpretation. Do let me know if you think that the contrast isn't good enough between the curves. Try to make it a little bit clearer. If anyone thinks this could be a source of confusion, do let me know. Next, we get to a figure where, I don't know, Jerome, if we should dive into this, but basically we had looked at these two figures. So one of them classifies several papers, A by scale. So the axes are scale, multi-area, single cell, and then by sort of mechanisms that are these clouds. And we do the same thing, but for OpenScope projects. And upon reviewing this, it did occur to us that what we're trying to do is fairly complex, and it is very interpretative. So we decided to make a table in which we would just write down exactly what we thought should be the categorization for every paper. And then Jerome has got in and added an extra layer of detail. And at this point, I'm not sure. I have several uncertainties about this figure. I don't know if I should dive into that, Jerome.
Yeah, no, I think we should continue that discussion in the GitHub forum.
Yeah.
Yeah.
Just really quick. While I'm totally on board and I really love the table, I thought that figure is the single most valuable figure of the whole review. So I feel quite strongly about it, even though there's a fudge factor. I think that is what all the genius of the review lies in. But yeah, we can discuss offline.
It would be really great then. I'd like to hear more from you, because the idea of the table was just to make sure that we weren't positioning things randomly, but that we had a grounded reason for where they were. I didn't necessarily think we would replace it with a table.
Just really quick. So I think the whole review and even this table can be done by ChatGPT or a future version of that. This figure cannot. There's this whole human element of understanding something very simply by coming up with 3 dimensions. Somebody looks at the figure, immediately gets the point, and nobody will get that point looking at the table. So that there was a lopsided space. It's filled lopsided. Everybody immediately gets the message. So that's why I think there's real genius in this figure. My heart would be broken to see it go. So maybe we can have both.
Yeah, I would love to work on it, maybe in closer collaboration with you, since you really have a vision for it. And just to make sure that we are confident about where we're placing the papers and about the message getting through clearly.
Yeah, I think we should just be open and say that's subjective. I think that's totally fine. I mean, we can use LMMs and come up with different numbers for that to make it a little bit more objective. But I think it's totally fine in scientific papers to have something called visual inspection, or as I said, like there's an author consensus here of about 60 people. So, or again, point to the table and say, look, this is here is a little bit more quantitative. But I think that it's more important, call it a schema or a cartoon or a simplified whatever, right? So this is not a data figure, and we don't claim this is data. So, but it makes visually a very important point. I think the most important point of the whole review.
Yeah, maybe just to comment, I think one of the problems, I don't know, Colleen, if that was an issue for you, is that if you put a reference at some point, it's in principle, one publication could cover several concepts, and then it's a bit tricky where to put it. But I mean, if it's like this publication was pushing this direction, was introducing this concept or so, it might still work. I was just this was one issue I had with the figure, was that sometimes it's hard to really put a reference, to one category or so.
That's right.
Yeah, I think the main thing for me is I don't have a fantastic memory for papers. So I found it personally very stressful to position other people's papers. And I just wanted to make sure I wasn't moving one randomly. But if we have
Can we see
consensus,
the
I'm very happy
table
with
again? Can we see the table?
that. Yeah. I figured if we had the table, I'd feel more confident
I
about
mean,
where I was positioning the
I
papers.
don't, like Alex said, I don't want to contradict what Alex said, but if I just want to share mine, I find it easier to follow the table than that. But it could be because I'm not seeing the thing that Alex is seeing there, and I haven't paid careful attention. But this is more linear for me to follow.
So what's the one shot sentence message you see in this table?
I thought the message was to know what papers are covering what topics and mechanisms, and that I can see easily. Like I see one line, I see what it's been marked for and what mechanism. So that I see it much easier here than there. But I see if your point is more for that categorization, maybe.
Well, it's also how the space is filled. It's how the space is filled, right? So it's not symmetrical. And as you see this right away in the table, I don't see it at all. In the figure, I see it right away.
I think this is the point of the figure is, I think as Alex said, is highlight that the literature is covering isolated corner of a phenomenon that is inclusive of all.
Meaning that we don't have one that sits, we have very few that sit in the center.
But well, for one, you could say that, but also like this is just like people are just thinking local mechanisms that implement certain things, and then this is an emergent network that leverage the whole process to make predictions. And
well,
so these things
also,
are connected.
I don't see an overlap between the red and the green. And so I thought that's a very important part of this paper, right? So the red is kind of filling up a different part of the space than the green.
But is that
and
not
that's literally one of the
incidental?
first sentences of our abstract. That we say that there's a problem, that the theories go one way and the data goes another, and we want to fix that.
So then my comment would be that then the caption of this figure should be written in a way that makes it clear our point is to show the separation between those. It's not about providing a list of papers and what they're about, because for that the table makes more sense. For this point, this makes more sense. So I would just change the caption, and even the title.
It's totally subject. Yeah, I agree, and it's totally subjective, and we should totally expect that authors will say, you put my paper in the wrong place, right? So then maybe if we do the table, and we could, feed these papers into an LLM and say, help us, and, make it pseudo-objective to place these papers in this three-dimensional space, so we get less of the blame. But overall, the visual point here, we can even maybe remove the papers entirely and don't put the author names on there. Just say, we found these papers and we put them there. We can just call them Roman numeral 1 and Roman numeral 2. But the point visually to say there's a difference between what's theoretically done and described and what's experimentally done. And then on the right-hand side, what has been done by the Allen Institute or what we're trying to fill. That for me, that's the actual graphical summary of this paper.
I see what you're saying. Okay. Yes. Because when you made the earlier point about the red and the green, I mistook that for the dendritic computations and EI balance. I see that you were saying the theoretical versus experimental are covering different areas. There was so much information that I could glean from it that I had overlooked that, and that's an important point. Okay, I see now. And I just want to check how we decided. Do you guys remember how we decided which papers to include? Is this a little bit arbitrary?
Yeah.
I
So that
think
is-
being arbitrary with citations is never a very good idea. My experience is that it doesn't, but yeah, like reviewers don't like that usually.
Don't like what?
My experience is that it makes more sense to be strategic with citations than arbitrary, to say it in very plain terms.
It's not arbitrary in the sense that any paper was chosen. I think some of these are, yeah, they drew paper that should be here are the one that drew the thinking of the community. I think that's, yeah, anchoring points.
Okay.
So if some anchoring points are missing, we should have had it.
Yeah. If somebody wants to go through the review and see if we've missed any major ones, that would be very welcome. But I don't really have time at this point to.
Is it so? I actually wanted to ask this. So our paper is now in review, and it's very relevant to the jitter part of the manuscript. So is it okay to add a reference at this point? Are we fine with that?
We have not submitted the paper yet.
No, she's referring to her lab.
Sorry. Yeah. The paper that came out from our lab recently on bioarchive. So I'm saying, can we add that reference? It's very relevant to parts of the manuscript. So I'm asking if it is okay to add citations at this point, not to this figure. I'm not talking about this figure, just in general.
I don't see why not, but you will have to where does it sit?
It sits very well with the temporal part, because that's what the paper is about. I'm not talking about this figure, just to be clear. I'm talking about the paper to be cited in the review, not this figure.
Oh yeah. You can. You definitely can add your reference. Yeah.
Because I thought the manuscript is now locked. So I mean, I can write you later about this. Not.
Oh no, we can edit the content at this point because we're changing the figures.
Yeah.
Okay, it's 10 minus 1. Colin, do you want to finish the last?
Yes, and then yeah.
And then we can continue the conversation on GitHub forum. I think Lucas, we might have to. Are you fine with presenting next week? Okay.
All right, I will go through quickly. This figure was just reorganized, and some of the excess colors were removed. We have the 3 types of sessions that share one oddball type, and then the other type that shares another oddball type, and then our shared control blocks. So I think this has helped quite a bit with the organization. The NeurPixel figure will be kept almost as is, with just a little visual fix. This was Kareem's figure, which we have kept almost as is. We did remove some colors for simplicity, and then we changed the colors here to match our updated colors from the figure that the original colors were borrowed from. So 10, 11 matches 13. The rest has been kept the same, just font changes. The next one, this change will have to be verified because we may have made changes that are not agreed upon. So it was partially a redrawing, but we were uncertain about the direction of the arrows from hypothesis to experiment. which were in the other direction in the original. So we want to double check that. And then we want to check whether this sort of use of the middle space and mirroring of the refine replace works now, where we sort of show that this applies to both the schema and an example of the schema in action. This figure was made originally by
Alex.
Alex, that's right. I don't know if Alex is here right now. Right, I stepped away.
Oh, he I guess he has another meeting. Yeah. Oh no, you're here.
No.
Yeah, they look good. Thank you.
Anything about, so the arrow direction, was that something that you think is very sort of important, or sort of doesn't matter which direction it goes in? I guess there's an argument for both, so. We thought it made sense because we were looping back through the red from experiment all the way back, so that's why we ended up deciding to change it. But yeah, definitely let us know if you think it's actually best the other way, and then we can shift where the red arrow is.
No, this looks
The
good.
next one is, okay, great. The next one was kept basically identical. This is the one that has a public domain image, which I've just mentioned in the caption now. And then we just have these two supplementals, Jerome, that we'll be needing the vectors for, and we'll just do some color harmonization. And those are the figures. So those are all the edits that we've made.
How do you do the color? How do you do the color harmonization? You've done a great job. Do you use software for that?
So yeah, we were doing everything in Illustrator, and then I think basically we decided on some color palettes, and Benedict made a lot of good choices for splitting into different colors. So Benedict is, I think, largely to thank for that. But yeah, fortunately, we had almost everything in vector format, making everything way easier. So yeah, the big figures still to discuss are clearly figure 8, 9, and figure 2, and it looks like for the rest, we're pretty much good to go. And thank you so much, everyone, for taking the time to go through all of this.
I wanna especially thank Colleen and Benedict for doing this over the Christmas break. Okay. So we'll continue that conversation on the GitHub forum. Let's reconvene next week with Lucas and a presentation on receptor field. We also have an invited speaker from NYU. Farzaneh, do you want to speak to that? Or?
Sure. Yeah. I don't have their name right in my head now, but I went to they're from NYU, from Shashin Wan's lab, if I pronounce the name correctly. I went to their poster at SFN, and they have a computational work on I can't remember, but related to
it's a proposal on how errors are computed. Yeah.
Exactly. Yeah.
And so it's in the agenda for next week.
Right. So it's a bioarchive, and anyway, they were interested in presenting to my lab, and then I thought this is a more appropriate venue for them to present, given the general interest. So yeah.
The name is John Meng from NYU.
Yeah. Yeah. Thank you. Yeah.
Okay. I'm sorry, Lucas, we can get to your piece. Please, anybody feel free to add comments on the GitHub forum. I'm actually gonna go through this in detail because it's important to us, and then we can start with that next week.
Perfect. Yeah. Jerome, just very quickly, can you just remind me again about your timeline with the?
That's right.
Because you wanted to go to production at some point in January, right?
In 2 to 3 weeks, we'll need to start. So that's why I'm probably going to provide comments in detail in the GitHub forum so that we can move on and make a decision next week.
Okay, perfect. Yep.
Anybody else has comment or question? Sorry we can get to your piece, Marcel and Lucas. I know you worked over the break. I really appreciate it.
Can I ask you a quick question, Jerome?
Yes.
To add the citation, is it now unlocked so I can just go to the Google Doc?
We'll send you a link to the piece
Okay.
that Colleen is working with me on.
Awesome. Thank you.
Thank you, Colleen, and everyone.
Thanks all. Bye.
Thank you. Bye.
See you. Bye.